Welcome to the Chemistry, Biology and Math Revision Hub. Today we are doing the A-level biology topics, and this is coordination in animals and plants. Beginning with topic 8b1, the central nervous system. The central nervous system is broken down into the brain and the spinal cord. So this is the brain and that is the spinal cord. And that's what we're going to be focusing on in the first part. The nervous system helps animals to respond to stimuli, and it consists of receptors, nerves, as well as effectors. The nervous system is broken down into the central nervous system as well as the peripheral nervous system. The spinal cord coordinates many unconscious reflex actions, as you will see a bit later. We begin by looking at the structure of the brain. The brain has so many different lobes. As we can see here, there is the temporal lobe, we have the frontal lobe, the parietal lobe, the occipital lobe, and so on. The cerebrum, or part of the cerebral cortex, is the largest part of the brain, and it contains two cerebral hemispheres. One is going to be to the left and the other is going to be to the right. This is involved in learning as well as critical thinking. It contains the corpus callosum, which connects the two cerebral hemispheres. This occipital lobe contains the visual cortex. The temporal lobe is for processing auditory information. The cerebellum coordinates movement of muscles as well as balance. The brain also contains a region called the medulla oblongata, and this is involved in processes like the heart rate, blood pressure, coughing, sneezing, peristalsis, breathing, and so many more. In the brain, we have the pituitary gland. This secretes hormones that control many body processes. Then the hypothalamus. This monitors the chemistry of the blood and controls the secretions of hormones from the pituitary gland. It is also involved in the regulatory processes like thermoregulation as well as osmoregulation. Here we have the structure of the spinal cord, which is another component of the central nervous system. It is made up of a core gray matter surrounded by white matter. This part here is gray matter, like we can see here, and is surrounded by white matter, which is due to the axons or the myelin sheath on the axons as they exit from the central nervous system. Impulses from the sensory receptors travel through the sensory neuron, through the dorsal root, towards the central nervous system. And impulses from the central nervous system travel through the motor nerve, through the ventral root, towards effectors, which are usually muscles or glands. We can see this is the ventral root, and that is the dorsal root. And here we have the dorsal root ganglion, which contains many cell bodies. Like I said already, the dorsal root contains only sensory nerve fibers that are going towards the central nervous system or towards the spinal cord, and then the ventral root carries motor nerve fibers from the spinal cord towards the specific effectors. Within the spinal cord, we have a relay neuron which synapses with both the sensory neuron as well as the motor neuron. The spinal cord coordinates reflex actions, and these include the rapid movement of a hand or foot away from a sharp or hard object, the swallowing of food, blinking of an eye, contraction and dilation of the muscles of the pupil, and so on. Next, we look at the spinal versus the cranial reflexes. Spinal reflexes involve the spinal cord directly, while cranial reflexes involve the brain. Examples of spinal reflexes include withdrawal of a hand from a hard or sharp object, while cranial reflexes include blinking, as well as the pupil reflexes. The pupil reflex involves the muscles of the iris, as we can see here. Here the pupil is wider or dilated, and here we see the pupil is constricted. These muscles include the saccular muscles as well as the radial muscles. When there is bright light, the saccular muscles are going to contract. The radial muscles will relax so that the pupil can constrict, as we can see here. And in dim light, the radial muscles are going to contract, the saccular muscles are going to relax, and the pupil is going to dilate in order to allow more light into the eyes so that clear vision can be achieved. Now, this is an example of the pupil reflex from your textbook. Here we see that light may enter one eye, or it could enter both eyes, it doesn't matter. The pupil from both eyes are going to respond the same way, because the cells of the retina are going to detect the stimuli and send impulses through the optic nerve to the brain. These impulses are going to be detected by the contrast center in the midbrain, and they will travel along two neurons to further contrast centers. As we can see here, this goes this way, and then it travels along that and that, or the other way around to further contrast centers, meaning there is going to be a connection to the next eye, even if light came from only one eye. So in these contrast centers, we see there is going to be synapsing with branches of the parasympathetic cranial nerve, and this is going to transmit the impulses to the iris. We see that going back to the iris as well as there to the iris. These impulses are going to stimulate the effectors, which are the muscles within the iris. Circular muscles are going to contract and the radial muscles are going to relax so the pupil can constrict. This is in case the eye was exposed to bright light. This is part of negative feedback that controls the diameter of the pupil in response to different intensities of light. The reflex response of either eye controls the dilation or constriction of the pupil in both eyes. So a bright light, which is shown on one eye, is going to cause the same effect in the other eye, even if the eye did not receive the same amount of light. Going on to topic 8b2, the peripheral nervous system. The peripheral nervous system is broken down or divided into sensory nerves as well as motor nerves. We know all sensory nerves within the peripheral nervous system are going to act the same way because they just get impulses from the receptor towards the central nervous system. However, these motor nerves are going to be different because they are divided into those that are voluntary and others that are involuntary. The voluntary involve the conscious thought of the brain, and the involuntary occur without conscious thought. The voluntary include working, talking, and dancing. You need to make a decision. And the involuntary can include taking away your hand from a hard or sharp object. So basically, these motor nerves are going to be subdivided because they respond to different situations. Some involve a reflex action, while others did not necessarily involve one. Also, the autonomous nervous system is going to be subdivided into the sympathetic as well as the parasympathetic nervous system. We can compare the sympathetic and the parasympathetic nervous system in terms of structure as well as function. You need to remember that these work antagonistically, where one causes an increase in intensity while the other slows down the effect. When we compare the structure, both have myelinated preganglionic fibers. We can see before the ganglia, both have myelin. And the postganglionic fibers do not have myelin, as we can see here. There is no myelin sheath on those. In the sympathetic nervous system, the preganglionic fibers are shorter, while the postganglionic fibers are longer. And in the parasympathetic system, the preganglionic fibers are longer, while the postganglionic fibers are shorter. Comparison of their function, the sympathetic nervous system is responsible for the fight or flight response, where it produces noradrenaline at the synapses and produces rapid response to target organs, while the parasympathetic system has a slowing and calming effect. Usually we call this the rest and digest, where it produces acetylcholine at the synapses. So you need to remember the similarities as well as differences in structure as well as function between the parasympathetic and the sympathetic nervous system. When we compare the nervous system and the chemical or hormonal control, chemical control is going to be slower and long-lasting, which makes it more effective in negative feedback systems. The nervous control is faster, short-lived, and it aids in quick response to specific stimuli. Going on to topic 8b3, investigating the brain. The brain can be investigated using CT scans. We can use MRI, functional MRI, as well as PET scans or positron emission tomography. So I'm beginning with the CT scans. CT scans use tiny beams of X-rays to image areas of the tissues like we can see here. There is an X-ray source here, another source here, and another source here, imaging different areas of the body. The density of the image tissue is going to affect the amount of X-rays that pass through. So denser tissues are going to reduce the X-rays, and the X-rays that pass through the system are going to be detected. You can see there is a detector below that. A computer system is going to produce a cross-section image of the tissue that has been imaged. CT scans can be used to detect brain tumors, brain bleeds, as well as swellings. However, they can't detect fine structure details. The images produced are going to be as photographs, and we cannot use it to detect changes during activity. The next one is MRI scans. In MRI, we use a magnet as well as radio waves or radio frequency. This is usually used to image soft tissues. During the process of scanning with an MRI machine, different tissues respond differently to the radio waves in the presence of a magnetic field. The hydrogen atoms within the tissues are going to be imaged, and they respond to radio waves in the presence of a magnetic field. A computer system is used to analyze the signal and produce an image. The images produced are going to be 2D scans. However, the computer can put together many slides to create a 3D image. These scans show more detail and they have less risk in comparison to the CT scans. Also, functional magnetic resonance imaging can be used. This makes it possible to watch the different areas of the brain in action while people perform different tasks. fMRI monitors the absorption of oxygen in different body tissues that are being examined. The deoxyhemoglobin absorbs radio wave signals and later re-emits it, while oxyhemoglobin does not. So we can see in areas where we have more deoxyhemoglobin, there will be more reabsorption in comparison to where we have more oxyhemoglobin. So blood flowing to active areas increases, and more oxyhemoglobin is going to be delivered to supply the active cells with oxygen for aerobic respiration. So less signal is going to be absorbed in those areas that are more active, and we can be able to detect them during activity. So an active area of the brain absorbs and so emits less energy than the less active areas. We can be able to make real-time observations in order to create images that can be interpreted using the fMRI machine. Next, we go to the PET scans, or the positron emission tomography. These produce 3D images of body parts. They show how well different parts of the body are working, and abnormalities can be detected clearly. You can combine them with other imaging techniques like CT scans as well as MRI to give a detailed diagnosis of a specific disease. The scans are produced by injecting a patient with a radioactive tracer. This could be similar to glucose, and the body is going to treat it as glucose. So it's going to be carried into cells which are more active. And since we know cells that are diseased like cancerous cells are going to be more active, more of the tracer is going to enter into them in comparison to the normal cells. So a scanner is going to be used to detect the radiation in the body, and a computer system is going to be used to analyze where it accumulates. Like I've said already, cancer areas or areas that are more active are going to take up more of the radioactive tracer, while inactive areas, like in cases of people with dementia, are going to take up less of a tracer, so we'll be able to tell the difference between the different regions of the body. Next, we go to topic 8b4, chemical balance of the brain. Here we're going to be talking about how neurotransmitter imbalances can lead to certain diseases. The first one we talk about Parkinson's disease. This is caused by the loss of dopamine-producing nerves in the substantia nigra area, and it's going to lead to gradual loss of motor control. The sufferers are going to experience symptoms like tremors, muscle rigidity, as well as slowed movement. Parkinson's disease can be treated using L-Dopa, which is a precursor for making dopamine. Dopamine cannot be given to the patients because it cannot cross the blood-brain barrier, but L-Dopa can cross the blood-brain barrier, so it's a good method for treatment of Parkinson's disease. Sometimes in treatment, they can use dopamine agonists. These are chemicals that bond to dopamine receptors. They have complementary shapes to dopamine, so they will bind to the receptors and mimic the effects of dopamine, meaning at the postsynaptic membrane there is going to be triggering of an action potential because receptors are going to bind to something that is similar to dopamine, and an impulse is going to be initiated. They can also use monoamine oxidase B inhibitors. These inhibitors are going to prevent the breakdown of dopamine by the enzyme monoamine oxidase B. So dopamine is going to be available in the synapses in order to trigger action potentials in the postsynaptic membrane. The next one is depression and serotonin. Serotonin is a neurotransmitter in many axons within the midbrain as well as the spinal cord. When the body lacks serotonin, there is going to be fewer nerve impulses traveling to the brain, and sometimes people are going to experience symptoms like depression. This can be treated using drugs or by using talking sessions to a specific therapist. The selective serotonin reuptake inhibitors, these are going to prevent the reuptake of serotonin back through the presynaptic membrane. So serotonin is going to remain in the synapse and more impulses are going to be produced in the postsynaptic neuron, creating a more positive mood. They can also use antidepressants. These increase the levels of serotonin and noradrenaline in the brain. In treatment with talking sessions, a therapist will help the patient or the sufferer to deal with the causes of depression, and this could occur without taking any drugs. Illegal drugs can also affect the brain. These include ecstasy, which is a stimulant that increases the heart rate. Ecstasy is a psychotic drug that alters the way a person sees the world. It affects processes like thermoregulation. It leads to raised blood pressure. It leads to irregular heartbeats. And it affects the hypothalamus, leading it to secrete more ADH hormone than normal. If the person who has taken the drug continues to drink more water, yet ADH is released in high amounts and more water is being retained, it could lead to destruction of more cells, due to the excessive uptake of water within the cells. So basically the cells could burst, and this could lead to death if the cells are involved in major organs. Next we go to topic AB5, chemical control systems in plants. Plants respond to chemicals. These are usually growth regulators. These control the growth as well as the development in plants. Sometimes they're called plant hormones. They are produced in one area of the plant and they could be transported throughout the whole plant to cause an effect in another region within that same plant. Some plant chemicals stimulate while others cause the inhibition. Growth responses in plant include tropisms, which are directional responses. Others include development as well as differentiation. So let's see how plants grow. Plants grow by cell division. So as cell divides, new cells are going to be created, but these cells need to take up more nutrients so that they can increase in size. So after the region of cell division, we see the region of assimilation, where the cells are taking up more nutrients in order to create more components within the cell. And as a result of that, there is going to be an increase in size, so we see the region of cell elongation. Elongation occurs, everything is going to be created. We can see now the vacuoles are created and many other cell components until a region of cell maturation is reached. Chemicals involved in growth include auxins. An example is indolacetic acid, which is a growth stimulant involved in apical dominance by suppressing the growth of lateral shoots. This enables the stem to grow fastest upwards instead of the lateral shoots growing. These are very effective in low concentrations. They are produced by young shoots and move downwards via active transport. If a tip of a stem is removed, root growth could slow down or it could stop. Auxins are involved in tropical responses, like in response to unilateral light. We're going to talk about that later. So let's see how auxins cause cell elongation. You remember we talked about the region of cell division, cell elongation, and then cell maturation. In this zone of cell division, the auxins are going to be synthesized. Meristem regions are regions where there is active cell division. And then these auxins are going to go downwards or diffuse from the tip or away from the tip. They are going to bind onto receptors on the cell surface membrane, activating the active pumping of hydrogen ions into the cell wall spaces. So the hydrogen ions are going to change the pH, creating an optimum pH, which is around 5 for some enzymes to function. The enzymes are going to break the bonds between the microfibules. This allows the microfibules to slide past each other, and so more water is going to be absorbed by osmosis. And due to tugger pressure, the more flexible cell walls are now going to stretch, increasing or expanding in size, and that leads to elongation. As these cells mature, the indolacetic acid or the auxins are going to be destroyed, the pH of the cell walls is going to rise, and the enzymes are going to be inhibited because the pH will not be optimum. So the microfibules are going to form the bonds, and then the cell wall is going to become more rigid. So that is when we've reached a region of cell maturation. The growth response example we have here is response to unidirectional light. When plants are exposed to more light in one direction in comparison to the other, light seems to cause the auxins to move laterally across the shoot, meaning it's going to concentrate on one region in comparison to the other. So there is going to be more auxin concentrating here. And since auxins like IAA are involved in elongation of the cells, we will see more elongation on this side. And this side is going to have less elongation. So the plant is going to grow in that manner, elongated on one side, and it will bend towards the light. Next we go to gibberellin and seed germination. Gibberellins act as plant hormones in many ways. One is as growth regulators. They stimulate the elongation of internodes as well as promotion of growth of fruits. In germination, they are involved in breaking seed dormancy. They do that by stimulating the production of amylase, and amylase is going to break down the starch from the starch stores in order to provide ATP needed for the processes involved around germination. So here we see a seed which absorbs water, and when water is absorbed, the embryo is going to be activated. Remember, for germination to occur, we need oxygen, we need the right temperature, and we need water. So water causes the swelling in order for the embryo to be activated. Now the embryo is going to secrete gibberellin that they fuse into the oloron layer. The gibberellin is going to stimulate that layer to produce amylase, which is a starch digesting enzyme. The amylase is going to break down the endosperm. Remember, the endosperm contains starch, so starch is going to be broken down into simple sugars that can be respired by the plant in order to provide energy or ATP required to facilitate the process of development of the shoot as well as the root. So we can see here finally germination has occurred. In topic 8b6, phytochrome and flowering. These phytochromes are photosensitive pigments that are found in plants. They exist in two forms. There is phytochrome red, which we sometimes call PR, and phytochrome far red, which we call PFR. PR is biologically inactive, while PFR is biologically active. We'll talk about this a little bit later. So when PR absorbs red light, it's going to be converted into PFR, and when PFR absorbs far red light, it's going to be converted into PR. So basically, they are interconverted into each other. The ratio of PR and PFR in a plant determines what time of day it is. So basically, plants can detect, or they can be able to find out what time of the day it is due to the ratio of the concentration of the two. As seedlings germinate, there is only PR because phytochrome is initially made as phytochrome red. However, as soon as the seed breaks through the surface of the soil and is exposed to red light, the phytochrome red is going to absorb the red light and become converted into phytochrome far red. So after that, we're going to have both PR and PFR existing in that specific plant. In the dark, PFR is going to be converted to PR very slowly, but PR will not be converted back. Also, PR is the most stable form of the pigment. Like I already said, PFR is biologically active, and it affects plant metabolism, even flowering patterns. Sunlight contains more red light in comparison to far red light, so during the day, most of the phytochrome in the plant is going to be far red or in the PFR form because red light is going to be absorbed by PR to be converted into PFR. And if the night period is long, then all the phytochrome is converted back into PR. Phytochromes affect plant flowering. Plant flowering depends on the length of day. The plants that flower when days are short and nights are longer become known as short day plants, or sometimes called SDPs. These include rice and cotton. And those that flower when the days are longer and the nights are short are called long day plants. These include oats and cabbage. Some plants are going to be called day neutral plants because they're not affected by the length of day, or flowering is not affected by the length of day. Also in the tropics, where the day length and the night length are almost equal or almost the same every day, flowering is not going to be affected by these chemicals, but it could be affected by something else, maybe presence of water and some other resource within the soil. In the short day plants, PFR will inhibit flowering, while in the long day plants, high PFR causes flowering. So in the exam, expect a lot of case studies. Maybe an experiment was carried out. Some plants were exposed to far red light and others to red light, flowering occurred, and so on. So data could be given to you and you're asked to interpret and be able to answer questions that are connected to that. Next we go to topic 8b7, phytochrome and transcription. During germination, etiolation can occur. This is when there is rapid lengthening of the stem, but less thickening. There is going to be less root growth, no leaves, and there will be no chlorophyll. This is like a mechanism allowing or enabling the plants to grow further in order to access light so that they can begin to photosynthesize. But after etiolation, meaning when they have access to light, elongation of the stems is going to slow down, the stems are going to become straight, and then leaves are going to gain chlorophyll so that photosynthesis can begin. Remember, we initially said phytochrome is synthesized as PR, and so these seeds are going to have PR only under the ground. This PR is going to be converted to PFR when exposed to red light, and after that there will be both versions. But also you need to remember PFR is the biologically active one. So since this part is about phytochromes acting as transcription factors, we're going to focus on phytochrome far red. So some studies were carried out, and again, you do not necessarily have to memorize the whole thing. All you need to know is there is evidence that PFR can act as a transcription factor by switching on specific genes involved in growth of plants. From the experiment carried out, the PFR moves into the nucleus and binds to a specific protein called phytochrome interacting factor 3. A complex is going to be formed between the two, and this complex is involved in controlling gene expression. We can see here phytochrome red interconverted into phytochrome far red. This phytochrome far red will enter into the nucleus. Inside the nucleus is going to bind to PIF3, and together you can see the complex is going to bind onto specific genes, causing the transcription of specific genes and therefore the production of specific proteins. Those proteins could be growth factors, growth regulators, they could be hormones that stimulate growth, and therefore specific chemicals are going to be produced that stimulate different processes that occur within the plants. It could be in fruit ripening, it could be in flowering, and so on. So this brings us to the end of this topic, which is about coordination in animals and plants. Thank you for being with us. Do not forget to subscribe. See you in the next video. Bye-bye.